Hello, I'm Beth McMillan and I'm here to talk to you about my PhD research. I work in the Department of Computer Science at the University of Oxford, and my research involves using mathematical models of the human heart to test drugs. I'm going to start by telling you a little about the heart and how it can be modelled, and then I'm going to talk about what that means for drug testing. The order in which the chambers of the heart contract is vital to its function pumping blood around the body, and this order is maintained by the electrical activity of the heart. You can see in this slide the grey heart tissue, and the red lines which signify electrical excitation. The blue line underneath is an electrocardiogram, which you might have seen on a heart monitor before. You can see that the electrical stimulation happens just before the tissue contracts. In each individual heart cell, when the cell is stimulated, its voltage goes up and then down again. This sets off a chain of events that makes the cell contract. The electrical system inside the heart is vital to its functioning. It's also surprisingly simple to model. To explain this idea, we need to think about what electricity is. Electricity is a flow of charge. Along a wire, charge is carried by electrons, but in biology, charge is carried by ions dissolved in liquid. So elements like chlorine, sodium, potassium, and calcium flowing in and out of the cell create currents. This can be represented very simply by the equation in this slide. What this says is that the change in the cell's voltage over time is equal to the sum of all the ion currents divided by the membrane's capacitance. There are some other more complicated equations behind this as well. People have been modelling the heart for over 50 years now, so the models are quite accurate. What does this mean for drug testing? When a new drug is invented for any illness, they have to check that it won't affect other parts of the body in an adverse way. Unfortunately, the ion channels that control the heart's electrical activity really like to bind to drugs. It's quite easy to measure how drugs will affect the individual ion channels of the heart, but because there are so many different ion currents, the way that a drug will affect heart rhythm is non-linear and quite difficult to predict. Changing the heart's rhythm can cause anything from mild discomfort to sudden death, which is a bad thing. Currently, one of the major ways that drugs are tested for safety is to either administer them to a live animal and see what happens, or to take some animal tissue and drop the drug onto it and see what happens. But these animal models can only catch around 70% of the drugs that cause arrhythmias. Can we use modelling of the heart cells as a better way of predicting arrhythmias? The answer is, quite possibly. If we can take data from single ion channel screens of drugs and plug it into the heart models, it might be possible to categorise drugs into levels of risk. This graph is from a paper some people in my department wrote that shows simulation results plotted against drug risk categories. If we include other model parameters, we might be able to refine this method even further. So essentially that's what I'm doing for the next 2.5 years of my life. And if it weren't for advances in computers and computing, then none of this would be possible.